The overall goal of this protocol is to describe a polyethylene glycol and zinc oxide templating method to create a porous ultrathin biomimetic basement membrane that supports dual cell culture and has tunable mechanical and chemical properties.
The main advantage of this technique is that we can create hydrogels that are ultra-thin, porated, mechanically, and biochemically tunable. They allow support of either single or dual cell culture. This method can help answer key questions in the tissue engineering field, including how dual cell cultures react to and interact with microenvironments that mimic human basement membranes.
The implications of this technique extend toward a wide variety of biological systems and tissues, ultimately allowing more biomimetic research with enhanced translation for human disease.
Add 250 milliliters of zinc nitrate to a flask and begin stirring the reaction. Then, add 150 milliliters of sodium hydroxide solution. A white precipitate will form briefly and then disappear as the two solutions continue to mix. Stir the mixture for 2 hours, uncovered. Heat the flask to 55 to 60 degrees Celsius and continue stirring for 24 hours. Turn off the heat and allow solution to cool to room temperature while stirring. Filter the solution through a Buchner funnel with an 11-micron pore size and allow it to dry overnight uncovered. When the filter paper is no longer wet from the poured solution and a white powder has formed over the funnel holes, the particles are fully dried. After checking the needle-like morphology of zinc oxide needle,